Okay, some thoughts on the metabolism of toxins to sort of complete the section on plant toxins, and we will loop back to acrylamide. That's what we're going to be looking at. Range of topics, including an interesting example of new product development right at the end. Okay, some definitions first, including the general category of exogens. But first we'll get some nonsense out of the way. The liver is the main organ of detoxification. Detoxification diets are a load of rubbish. Okay, so exogens, substances originating or produced outside of the organism, compared to endogens, which are those that derive from normal metabolic processes. Exogens include drugs, but in this context we're interested in products produced from normal sort of process in food. Now we will talk a little bit about drugs as we go through this particular section. Metabolism mainly takes place in the liver, with the kidney, skin and a few other organs being involved. The chemistry involved often acts to increase the polarity of these exogens, so making them easy to excrete via the kidneys. Put more of a charge on them, really. Usually reduces toxicity. There are some famous cases which aren't covered here, which do increase toxicity. Beta carotene and smokers is one of the classic examples which you might want to look up yourself. And it occurs in something called the hepatic microsomal enzyme system. And here's some pictures of it. Many reactions take place in the MES, which is called the smooth endoplasmic reticulum. That is the endoplasmic reticulum, which doesn't have ribosomes studied into it. Connective tissue throughout cells, in this case particularly in the liver, is what we're interested in. Two types of reactions take place. Type 1 reactions add or change functional groups, and we'll look at one of them a little bit later on, while type 2 increase molar mass. In both cases, as mentioned, they act to increase polarity. Here's an example from drug discovery. Willow bark was traditionally chewed to act as a painkiller. It contains acetamilide, which is an analgesic. Early in the last century, paracetamol, a more effective painkiller, was found in the urine of people who chewed willow bark. And the conversion of acetamilide to paracetamol is an example of a type 1 reaction, with the addition of an OH group to acetamilide, so making it slightly more soluble in water and more polar. So, what about the enzymes that do all these processes? These are cytochrome P450s. Why the liver is a red-brown colour. Cytochromes are widely distributed in animal biology. Three broad categories, and the numbers there are sort of absorption bands, and they're all sort of in the red area. We're not going to go into the details of the various different ones here. We're just going to treat them as a general category. There are porphyrins, which are very common in both animal and plant biology. P450s have a porphyrin ring with iron as a metal involved in the oxidation reactions they catalyse. Here's some other examples. Chloroform with magnesium, heme again with iron, and then cytochrome P450 with iron again at the centre, the so-called heme group. They're involved in lots of other roles, not just drug metabolism or exogen metabolism. They have key roles in cholesterol synthesis, vitamin D metabolism, and with the electron transport chain, which I'm sure you remember from human metabolism. So drugs and exogens in general are often metabolized by P450s. P450 is involved in the metabolism and ultimately bioinactivation of 50% of all drugs, so they are quite important in pharmacology. Here's a potential problem. Grape juice is associated with the inhibition of cytochrome P450s, particularly CYP34A enzyme. If you want to have a read of it, read the NHS link that's linked in the notes. Also found it also seems to have a problem with marmalades as well. Pomelo is a citrus fruit native to Southeast Asia. Citrus maximus was originally called shaddock in England after captain of an East India ship company introduced it in Jamaica in 1696. It's a bit like a grapefruit, I guess. Okay, return to acrylamide. This paper is a good read for anyone wanting a deep read into acrylamide metabolism and toxicity. I've just quoted some examples of acrylamide from the paper. Acrylamide concentrations, again, quite a wide range, except for infant powdered formula, which is obviously a highly produced industrial product. Okay, a flashback. A reminder of the Maillard reaction, the production of acrylamide. The acrylamide molecule is small and hydrophobic, and it easily diffuses through tissues. If you may recall from biochemistry, passive diffusion is diffusion that doesn't require any energy. It just happens across membranes naturally. Many gases, for example, will diffuse in and out of cells, and so will acrylamide. This is a reaction that forms the genotoxic glycinamide, and it's catalysed by the CYP2E1 variety of cytochrome P450. And it goes on to cause potentially a range of damages. It's quite an unusual looking molecule. A little triangle in the corner with the oxygen in one corner suggests it's going to be pretty reactive to any chemist. And it does, it binds to DNA, producing what are called stable DNA adducts, that is to say, molecules which bind firmly to DNA, potentially producing genotoxic effects and maybe leading to carcinogenesis. DNA adducts are a form of DNA damage, as I said, caused by covalent detection of a chemical, MOTI, it says in the notes, that is a molecule, basically the DNA. If they're not removed by the cell, they can cause mutations which may, for example, give rise to cancer or other conditions. They're frequently used as biomarkers for chemical hazard exposure or cancer therapy efficacy. And from our point of view, at least 6% of ingested acrylamide is converted to glycinamide. And again, there's a link there to an article from Nature if you want to do a little bit more reading on DNA adducts. Okay, kinesin proteins, which again can be damaged. There's a really great video there, though it talks a little bit about this being intelligent design at the end. It is not intelligent design, it is evolution. Okay, so kinesins are proteins belonging to a class of motor proteins found in eukaryotes. They move around microtubule filaments and are powered by the hydrolysis of ATP. What they basically do is move structures within cells. And they say it's a really nice video, apart from the rather strange ending. So have a look at the video, which is linked in the notes as usual, and I'll also post it into the YouTube thing as well if I have a minute. Okay, kinesins are also involved in the formation of spindle proteins. Now, spindle proteins form when genetic material divides in a cell. I'm not going to get details of this. We probably covered this in cell biology in first year. The spindle is necessary to equally divide the chromosomes in the parental cell, for example, into two daughter cells, during which both types of nuclear division, mitosis and meiosis, are involved. And again, acrylamide may interfere with the kinesins involved in this, so it may affect cell proliferation with a range of potentially negative effects. Okay, here's a possible way to mitigate the potential negative effects of acrylamide. Lactic acid bacteria, probiotic organisms, appear to demonstrate the ability to bind acrylamide and use it as a carbon source. It's not going to be involved in any other actions, it's just going to be rendered down for energy. This may potentially lead to acrylamide toxicity. Again, see the table in Kazuka et al., the paper we had a little reference to a little bit earlier on. And finally, I look forward to the second semester module where we'll be looking at innovative product developments in food. And no, this really is a product. A Singaporean ice supplier, Unitat Ice, and marketing created probiotic ice cubes. The ice cubes consist of filtered water and bifidobacterium, up to 2.8 billion organisms per cube. Whether they're still active is an interesting question, I guess. Under proper storage conditions, the people who marketed this product claim the products can remain viable up to 6 months. So what do you think of that product? Okay, that's all I want to say about that. Thanks for listening.